So please, as I'm going into this, feel free to interrupt and feel free to, ask questions and challenge stuff that I say if you think it's wrong or incomplete. And I think particularly I'm approaching this from the angle of someone who works on empirical systems and who is interested in things about work. And the only difference maybe is that I've been doing it slightly longer, and I've been frustrated as I've worked on these sorts of systems and how we approach the questions. So some of this will be a little bit of a sort of maybe a tour of my brain as I've changed my mind over the years. So is the microphone on? Can people hear me? So apparently speakers are asked to use the microphone so that we have a good audio of this when it's posted online. Okay, so we're all familiar with phylogeography, and we're all, I think, particularly used to the way of thinking about it that was introduced by John Avis and his colleagues. And Avis was trained as a systematist. He certainly knew a lot about population genetics, but his background was a background of systematics. This is something that many of us have in common. And he was very interested in this idea that we could use phylogenetic thinking to ask questions about populations and the beginning stages of speciation. And so if you read his papers or this book that was published in the year 2000, how many people have read some or all of this book? Probably many of us, right? So, he defines phylogeography, and he does a really good job of talking about why it matters and what it's interested in. And basically, he defines it as being interested in understanding the principles and processes that influence the geographical distribution of variation, right? And so he's interested in these two axes, time and space. And he's trying to understand something essentially about the history of genetic variation in these populations. And I think the reason that what Avis did was exciting and new to people is that he wasn't just interested in characterizing genetic variation in the present. He very much wanted to understand how it was formed. And so it's overtly historical as a discipline. And many people have sort of attributed Avis and phylogeography in forming a bridge between population genetics and more phylogenetic types of approaches. And in fact, Avis promoted this idea and titled a review paper that was sort of a very seminal piece of work, this mitochondrial bridge between popgen and systematics. I think this emphasizes a second aspect of the beginnings of phylogeography that we've moved away from. It's a systematic tree-based approach and relies on mitochondrial DNA, at least in the beginning stages. Even today, I would argue that most of us, especially if we're working in animals, the first gene we're going to sequence is probably site B or CO1 or something like that. So how many people have started working on a new system and done this? I mean, okay, so it's pretty common, right? And we don't stop there now. The trouble with the reason that poor people like Stu have been in PhD programs so long is when I was a PhD student, you could stop at site B, and everyone was happy about that. And the world changed, and that's good for me, I guess, and bad for Stu and everyone else. But the reason that mite DNA was promoted is that it had a high mutation rate, these small effective population sizes. It formed phylogenetic patterns. People could look at these trees on a map, and they could make inferences about them. And so for years, I've been showing this figure in talks, which is basically, phylogeography in the era of Avis and his colleagues was estimating a gene tree from a gene like site B, putting it on a map. And at this point, you might have really rigorous ways of collecting the data and making the estimate of the gene tree. There was talk about models of sequence evolution and whether you should use certain kinds of heuristic searches or Bayesian searches or whatever. But once you get here, all of the inferences that are made are really qualitative, right? And so you might tell essentially some narrative, some story about how there's a region that's causing, inhospitable or something. There's no gene flow there, and that's why you have the separation of whatever species this is into a southern clade and a northern clade, for example. And so as phylogeography has developed in the last 10 or 15 years, we've gone from this really qualitative storytelling approach to the science to one that is increasingly concerned with being rigorous, with making statistical inferences, with using models to estimate parameters. And this is undoubtedly a really good thing. But what I want to ask you and what I would like you to think about is the process of inference itself. When we go from what results we get from our data to understanding something about the demography of the species that we're interested in, how much has that become quantitative? And how have we really gotten away from this sort of qualitative narrative approach to phylogeography? And I want to talk about a couple of papers as a way, and really have a discussion about these. These are in the shared folder. We've read these. And not to pick on these papers or anything, because I think both of them are really good papers, and we can read them today, and it's important to recognize that when they were written, they were very much at the leading edge, important work that lots of people read and thought about and talked about. And so the first one is this paper by Marjorie Matak, and it's dealing with these wood rats. And I put this up, the slide up here in this little paragraph, because it really emphasizes what the goals of this research are. And in many ways, these goals would be the same if she was going to repeat this study today with RADseq data or something. So take a second and read this paragraph. So what are the goals of this study? This is right out of the introduction. So what's the motivation here? Do we need to read it again? Yeah. Come on, Stu, help me out. I'm going to pick on you today.
Yeah, to place the mitochondrial history into a geographic historical context.
Okay, so
one goal. Anyway.
Understanding something about the genetic diversity, which is being equated here with the mitochondrial DNA. It's the gene that's being sequenced. Placing it into a historical context. Understanding something about climate change during the Pleistocene, essentially. And so as goals, for phylogeography, even though this paper is 15 years old, we could imagine many papers that would be written today, including in many of our labs, that have the same basic approaches. And so here's sampling localities, and most of the work in this field has done a really good job at sampling broadly throughout the range of species. This is seen as one of the important goals, and I think it's something that, clearly this paper exemplifies. And so the approach, the methodological approach used in this paper is to make summaries of genetic variation, right? And so in the materials and methods, things like FSTs are calculated. Tajima's D statistic is calculated. These are basic measurements of genetic variation. A gene tree is estimated from the data directly. And then these results are interpreted. And so we see sentences like these, phrases like these in the results section and the discussion section. So Tajima's D, these values are not statistically significant, but there's a negative value in the north that Marjorie interprets as possibly being an indication of a bottleneck. So we're trying to make inferences based on patterns in the data, right? For the gene trees, there's a little more information there. The geographical distribution of distinct clades suggests that there's some combination of these topographic barriers, expansion and contraction of suitable habitat, during this time period, and it's looked at as being coincident with these mountain ranges. And so here we have the sort of the main inferences that are resulting from this work, and there are inferences that are based on looking at the pattern of variation in the genealogy and comparing it to the geography, right? And so phylogeography studies often have these kinds of figures in them. And the variation, the mountain ranges are not shown on here, but unless people who have done work in this area have this understanding that certain ranges seem to influence genetic diversity, this seems to fit that pattern. Okay. And so I would argue that this is a nicely done representative paper from this sort of early stage of phylogeographic research. when you read it or reread it, anything about it, was anything striking or seemed missing or absent?
I mean, any other hypotheses for why these lineages would have been distributed like this? They just sort of like throw out climate oscillations of whatever time period that was.
Where were those oscillations discussed, and where was that aspect of it discussed in the paper itself? I mean, was it proposed?
I don't think it was set up as like hypothesis, really. It was more like they just noticed destruction, were like sort of arm wavy. Maybe it's climate.
Okay. I mean, it certainly doesn't strike you when you read it as a paper that's designed to overtly test hypotheses. And this is, again, fairly common for these kinds of early studies. I think it's worth debating and something that we should talk about, in 10 minutes or so, if that's necessarily the end-all, be-all of the kind of work that we do. But, this work is more focused on interpreting the pattern in sort of a post hoc way. And again, this is, state of the art for when it's written. One of the things I really like about this paper, it's actually one of the first papers to use a coalescent model to estimate population level parameters. And so it uses a really early version of fluctuate. This should say theta up here. Something happened, or I didn't use the right font. But they use Mary Kuhner's program fluctuate to estimate theta and this parameter gamma, which is a measure of the extrinsic rate of population expansion. And I think it's one of the papers that's being written at the first stages of the implementation of these coalescent models in phylogeography. And in the discussion, this is commented on, these positive growth values indicate that there are ongoing and or recent increases in the population size in both the northern and southern regions. She uses a t-test to see whether growth is more rapid in the north and the south and sort of evaluate whether expansion has happened more in one area than the other. But it's definitely, I think, one of the reasons this is a really remarkable paper in a lot of ways is that it applies these really new methods to this system in a way that makes perfect sense. We can also look at it, it does something a little more conventional. Lots of people use molecular clocks to estimate population divergence, and so it does that for some of the clades in the tree as well. And this is looked at as an important step in the pattern of inference. So it's not the case that there's, this is not the tree on the map of Mordor and that's all. There's lots of other stuff going on here. Okay, any other comments about the Woodrat paper? And I think this sort of highlights a distinction. There's really two ways that we can ask questions of the genetic data that we're seeking to gather. One are these summaries that we can make of genetic variation, and so things like Tajima's D or FST, we can estimate a gene tree. There's also these population level parameters that we can estimate. And we talked about theta and gamma. There's these isolation with migration models that are that were introduced around this time that are also used by many phylogeographic studies. And so the next paper that I would like to talk about really focuses on these IM models. So this is work out of Craig Moritz's lab that was published in 2006. It's one of the early phylogeography papers that takes a multi-locus perspective. And it's been cited a number of times as an example of why one would want to collect data from more than just site B. So it's a really nice paper. Like a lot of Craig's work, they are interested in the Australian wet tropics. And they have these are lizards, right? Lizards that are distributed in different regions across known biogeographic barriers in the Australian wet tropics. And basically they have these three things they're interested in. Northern and southern populations of this species, rubricularis, and then a sister taxa, or what was initially thought as being a sister taxa, this rhomboidealis. And when they estimate relationships among their samples, they sort of have the complicated finding that this other species is actually forming a clade with half of what is described as the second species. And so This puts them in a bit of a difficult situation because they wanted to use this new model, new at the time model that's been introduced, IM, out of Jody Hayes' lab. It estimates isolation, population isolation, and migration rates between two diverging populations, right? And they have three populations of interest here. And so Dolman and Moritz do something that's entirely reasonable, and they take this three population question and they sort of divide it up into pairwise comparisons. And they first estimate divergence between these two groups, and secondarily, estimate divergence within this entity that's described as a single species to the exclusion of this group here. Okay. And when they do that, they make parameter estimates. And what I've done here is I've used, none of my thetas are going to show up as thetas here, but I've used just color coding to sort of highlight what people are getting, or what estimates they're getting from these programs. And these are theta values, so ancestral theta, either, I guess, of this clade, or this lineage, and then the thetas of these individual populations. And they're also, of course, jointly estimating population divergence and migration rates with this. And they then do what with these parameter estimates? So why did they go through all this trouble to do this?
See how distinct they were. To see how distinct the population was.
See how different they are temporally, understand something about when they were isolated from one another. So they're mainly interested in these values here for the population divergence. They're probably influenced, there's this well-known Beerley and Edwards paper that talks about estimating T and why you want to collect multi-locus data to get a better estimate of population divergence. This was really influential at this time, and lots of people had read it and were collecting these data to get these things. And so they're interested in population divergence. They're trying to understand something about the history of species, probably make inferences about the taxonomy, and this sort of a messy situation here. So one of the things that is worth noticing about this kind of situation is that when you estimate a parameter given one of these coalescent models, you never estimate anything independent of the other parameters in the model, right? And so these are all jointly estimated parameters. And you can have the same population, depending on how the data are analyzed, end up with different estimates of a parameter, not because the model isn't working well, but just because you're analyzing different data in comparison. And so the best example of this is both of these parameters are measuring genetic diversity in this red population. And depending on if you compare it and evaluate the ancestral diversity here or there, you'll get slightly different values. Now, they're not off by much, and so who knows how important this is. But the same genetic data in this red population are being analyzed in sort of two different comparative ways. You're getting slightly different answers. It's also, I think, it would be really interesting to take these data, and actually I've downloaded these data and they're running on this new IMA2 version because I wanted to compare these estimates to what would happen with a three population model, but I can't get the thing to converge. And so this is a problem that sometimes happens with IM, but I do want to see how estimating all of these simultaneously influences this as well. Okay. We've often, I think, when I started using these programs, I didn't appreciate how conditional parameter estimation was on the models that were being used and the comparisons that were being made. We have this idea that there's some true value out there and that we just need to estimate it, but there's subtle things that can influence our ability to estimate that value, and one of them is the composition of the model that we're using. This really was sort of driven home to me when my former postdoc, Maggie Koopman, and I used again, use this program, Lamarck, that Mary Cooner has been working on, to estimate theta and gene flow using a model that included population expansion and then a model that didn't have expansion in there. And so when we estimate theta, under the assumption that there has been no growth, we get a higher estimate. These are the two dots here are two different populations, east and west. If we assume growth, then all of a sudden we get a lower estimate. So why is that? Conceptually, why should our estimate of theta change so dramatically if we include this additional parameter?
The coalescent assumes a constant population size.
Well, so the model, if it assumes the constant population size, why are we getting a higher value?
You're sampling at the tips. You're sampling at the tips, and you are overestimating theta in the past. If it's in a coalescent framework. That's what I'm getting at.
So if we sample at the tips, are we overestimating theta, or we're just
If
why does that value get inflated?
The population grows exponentially, let's say, you'd overestimate theta at some time in the past.
Okay, so if we assume that the population is growing, a smaller value of theta sort of fits the data that we're putting into the program. Right, so we have some pattern of genetic variation. And if we assume growth, then the maximum likelihood or the highest probability of the data that we've collected will sort of settle at a lower value of theta because the expansion of the population is in part helping to explain the genetic variation that we've detected. Does that make sense? Laura, do you have a better explanation of that?
I think that works.
Laura has the unfortunate position of being the statistician that I ask questions. So we have a certain amount of variation in our data. And as we add parameters, there's more ways to explain the variation that we have, in essence. We see the same kind of thing with estimates of migration. Again, these are two populations, and so gene flow going into each of the populations. And if we assume growth, then all of a sudden a model where one of the populations has a lot more gene flow going into it is a much better fit for our data. So we have these relatively low migration rates, but as soon as we model growth with it, then we change our interpretation of how much migration is occurring into one of these populations. When we wrote this paper, this was disconcerting because we wanted to understand the relative contributions of population expansion and gene flow in these pitcher plants that we were working on. And it's hard when we look at results like this to make any sort of qualitative inference about how important these processes are. Right? We can say something about the estimates of the parameters, but unfortunately, there's no way to evaluate which of these models is a better fit for the data. Or there's no easy way to do that. And one of the reasons that we're here today is because it was this kind of result that got Brian and I talking about doing model selection in the phylogeographic framework, and led eventually to me talking Brian into doing the hard work of coding this so that we could actually evaluate it. Okay. It's the kind of thing that people and the people in this room, I think are like this. If you're curious about your empirical system, you don't analyze it in one way and stop and write the paper. Most of us analyze our data sets in many different ways, and we try to figure out which of these approaches are most reasonable or give us the most information or are most worth including in publications, right? But this is a hard thing to decide, and it's hard to decide after the fact. I have no idea which one of these models is better for the data that we put into it to this day, and so it's a difficult thing to evaluate. And to summarize where we're at without some evaluation of model fit, we can summarize genetic variation, we can estimate population genetic parameters in a number of ways. There's lots of formal approaches to doing this. There have been dozens of papers written about which models give you the best estimates of certain parameters under certain conditions, right? We've all seen the sort of simulation testing approach. But regardless of how good a model is at estimating the parameter that we're interested in, we are always interpreting and making inferences based on our sort of qualitative interpretation of these parameter values. Often we're assuming that model fit is good to the data that we're analyzing. You know, we all want to use programs like IM or Migrate because we can get answers. They're relatively easy to use. You know, reviewers expect that we're going to analyze our data in sophisticated ways, and these are among the most sophisticated ways that we can do it. But we're still developing our inferences based on sort of qualitative impressions of this. There's at least two reasons why we should, I think, be a little cautious about this kind of approach. One was pointed out by Knowles and Madison in the statistical phylogeography paper that they wrote in 2002. And they basically argue that most empiricists will develop a more detailed historical scenario than their data can really support. And I think this is something that I have done many times, and most of us are probably guilty of this. We work really hard to collect samples. You know, many of us travel to really difficult to access field localities. We get samples. We do everything we can to get the money to collect the data. It's not easy, of course, to do the lab work and to get good results. And when we finally do all of that, of course, we want to get as much information out of our data as possible. It's the natural sort of approach that one has. And it's hard, especially when there's no good way to evaluate if you're overanalyzing or overinterpreting. Of course you're going to do that. And you know, most reviewers encourage this. They want to see that sort of speculative discussion in many cases. The other problem, and these are very much related, is that there's a phenomenon that psychologists are aware of that's known as confirmation bias. And what this basically means is that as we get new information, we interpret it in a manner that's consistent with what we think we know already. So if we think we know something about the empirical systems that we're working in, every new bit of information that we get is going to be interpreted in light of what we think we know. I would argue that what we think we know about systems largely drives how we analyze our data. If we have two different things, population, species, whatever, and we think they're common ancestor, you know, they shared a common ancestor relatively recently, we're going to naturally think of these isolation models, maybe isolation with migration models. We might not even use a model like Fluctuate that might be perfectly good and maybe give us a better estimate of something because it's closer to the truth, right? So when we analyze our data, I think sometimes what we think we know can get in the way of making unbiased choices about even how to estimate the parameters that we're interested in. Of course, absent an approach to evaluating the fit of your models that you're using to your data, it's all speculative, and we could say this, and you know, what can you do about it? And again, one of the goals of FRAPL is to understand and to develop objective approaches to assessing the fit of different models to the data. And so we're going to talk a lot about this later in the workshop. Okay, so how should we analyze our data? If we really want to know how genetic diversity is partitioned across the landscape, and if we really want to identify demographic events, I would argue that being model-based is a first step towards doing this. And so models can be used to describe as complicated, as complex of a demographic history as we can imagine. And I think all of us have, when we've analyzed our data, we've sat down, and so, you know, I think a couple months ago, Teresa and I were in my office, and we're drawing pictures on paper, and oh, you know, if this happened, then it would be like this, and sort of we've sketched out models, and we've made sort of these interpretations of what we think might be going on in the systems that we're interested in, right? And so all we want to do is we want to make these formal, and we want to think about the different parameters that we're going to include in these models. And so things like population size, divergence time, size, population size change, gene flow, are all parameters that we could reasonably include in the models that we're interested in. We could, of course, put selection into this for certain genes, but FRAPL assumes, and I think for the standpoint of the historical demography that we're interested in, if we find genes under selection, we're probably going to ignore those while we try to understand something about the demographic history using the neutral loci, and then maybe we'll come back to the selection issue. So the good news is that for these neutral markers and these demographic history kinds of questions, there aren't that many parameters that one would really want to include in models. And so I think these are the four most important, and we're probably all in agreement about that. Now, the complex thing is that each of these parameters can either be used to, or be applied to a single population or something that's happening between populations, like gene flow. And so as our models become more complex because we add more populations to them, the scope of these models and the number of parameters that they include can increase pretty quickly. And what that also means is that when we think about model selection, as we increase the complexity of the systems that we're interested in, the number of models that we have to consider will increase dramatically as well. And so we will talk about how and why and how certain choices that you make can change the complexity of model space. But for now, it's enough to say, you know, if we have one population, it's really pretty easy to model it because we don't have to worry about gene flow. We don't have to worry about divergence time. You know, the question might be, is it growing or not? Has there been a bottleneck or not? As soon as we add two, then all of a sudden we can have these other parameters. If we have 3 or 4 or 5, then things can get very messy and complicated really quickly. So on average, how many, when people think about their data, how many populations in your empirical systems do you typically model or try to model? So, Jen, with the irises, about how many?
3 or 1.
3 or 1? Okay, so Katie, you said 2?
3.
A 3? Okay. That?
Probably 2.
Or 2. Anyone else?
As many,
2, 3? Good.
Yeah.
What a relief. So it turns out
as many as I can.
as many as I can. Okay. Well, there's always one. Well, your Frapple runs might last until the end of time. Hope you don't need Frapple to finish your dissertation. Okay. So there's a relationship between the complexity of the model and how long any kind of analysis takes, and so that's something that we're definitely seeing with Frapple. Okay. So I would argue that if we want to analyze data, one really good approach to doing it is to overtly try to assess model fit and try to understand how models can fit our empirical data. So these are 2 models that were developed by Lacey and I for a paper that we worked on while I was her postdoc. And these are grasshoppers, of course. You know, Lacey loves grasshoppers. But we basically came up with was a model where during the Pleistocene, all these populations were sort of in a single refuge, and then they expanded at the end of the Pleistocene. And so here's timing of that, versus one where they were separated into these eastern and western refugia, and then there was a expansion from each of those. And so we had these models, and we wanted to evaluate them overtly. And the question is, how are we going to do this? And how did we do it then? How might we do it differently now? And so in order to sort of think about model selection, what we need to do is think a little bit about the different statistical approaches that one can take to answering these questions. And so I'm going to talk about 3 different approaches and start with the one that is sort of traditional statistical approach that most of us are familiar with if we've had like a biostats sort of class, use a book like Sokol and Rolf. And this is something that is sometimes called frequentist statistics. Is that, Laura, is that a reasonable sort of characterization? So the goal here is to identify hypotheses that you cannot reject as false. And so if we had these 2 models, when we took this approach, we were trying to see if we could find a model that we couldn't reject, because that then would imply that the model was a reasonably good fit to our data. And so this is part of a really long tradition in biology of this null hypothesis testing. And basically, when we do it, we calculate the probability of an observation, so the data that we've collected, conditioned on the null hypothesis, and we tried to sort of evaluate if the probability of the observation is, you know, a reasonable thing given the null hypothesis or an extreme outlier given that null hypothesis. And so we've all seen these kinds of tests where you have some distribution, and if your empirical data are far outside of the distribution of your expected values, then this is indicative of the fact that your data are not a good fit to the model. You would reject this test. Typically, an alpha value is used as a cutoff. And so this is where the p equals 0.01 or 0.05 or whatever it is comes into it. Usually, this value is sort of thought of as being a boundary between either strong or weak evidence against some null hypothesis. Okay, so we've all done this sort of hypothesis test. I think, anything from looking at Hardy-Weinberg equilibrium using a chi-square test or any of a number of other ways, right? And so this is essentially a traditional, a relatively straightforward approach to hypothesis testing. One of the sort of more subtle goals statistically is to try to minimize the statistical error that you're introducing into these tests by thinking about both the type 1 and the type 2 error rates. And so the type 1 error is rejecting a null hypothesis that is actually true. The type 2 error is a false negative, or failing to reject a model that is actually false. It's worded poorly. For both of these, we need some distribution of values, and we need some cutoff that we're willing to tolerate. And so the level, the alpha level, is essentially the how you go about trying to minimize the type 1 error, the type 2 error. You're finding something that you're comfortable with. And so usually we might use 0.05 or something traditionally. Okay. So for phylogeographic research, the big question is, which empirical values should we use, and what statistical distributions are good to test against? And it becomes problematic because most of our data do not fit the assumptions of standard statistical distributions, right? So like chi-square distribution or like a standard normal distribution, what are some of the assumptions that when you take biostats, they tell you about for these kinds of things? Your data points should be what? They should be normal, which means what? Okay, so they have like sort of an expected level of deviance to the variance. The data points should be independent. And sort of, you're invoking certain assumptions that statisticians make about, that your data are sort of an independent sample of some true distribution, et cetera. And a lot of these things are not directly relevant to the kind of data that we collect. So for example, if we collect data from site B, you can't argue that each of the sites are independent because they're evolving along some chromosome that is very much conditional on what's happening at other parts of it, right? And so it's, I think, important to think about these models and to think about the distributions that we're using when we do this. Standard statistics is developed around this idea that parameters define the dimensionality or the complexity of model space, and the models describe these relationships. And so we've all done regressions. We've all sort of done tests where we're comparing linear models to parabolic models or something like this. The kind of statistics that we use when we do null hypothesis testing is based on these kinds of models and parameters. They're related to, in many cases, the models that we're using for phylogeography, but I don't think there's a direct one-to-one translation there. The way that the testing is done in sort of standard statistical approaches is also different. And so in this case, we have models that are nested in one another because we have a model that has, you can reduce the model on the right by what? By treating C as 0. So there are these nested models, and essentially when you do this Nyman-Pearson testing, you're trying to reject these, this additional complexity to see if you really need that second term to explain the data that you've collected. Again, these are not directly translatable to phylogeographic research. And so when we were trying to make phylogeographic research more quantitative and less reliant on these sort of qualitative interpretations, one of the ideas that people had was to take hypothesis testing approaches. And when this was thought about, the immediate realization was that we can't use necessarily the standard distributions, the standard ways of testing. And so what we need to do is be a little more creative with the test distributions that we get. Okay, so we were talking about parametric bootstraps and in the context of hypothesis testing. And what I was trying to convince you of during the fire alarm was basically that standard approaches to hypothesis testing are difficult to apply to phylogeography. But what people have done, and one way that we can use hypothesis testing in a frequentist manner, is to simulate our own null distributions via this process known as parametric bootstrapping. And so this is a statistical technique that allows us to build a null distribution of the probability of the parameter that we're interested in, given the assumption that the null hypothesis is true. And so basically, we're calculating the probability of our data, given that some null is a true model. And so by comparing our estimates of what we're interested in, we can evaluate the null hypothesis. This has been used in a couple of different ways for phylogeography. So I think Jack Sullivan is the first person to use parametric bootstrapping in phylogeography. And this 2000 paper, what Jack did is he used sort of a phylogenetic approach. He took the gene tree from one species of rodent and compared it to the pattern in the second species as a way to see if they had the same phylogeographic pattern or a different phylogeographic pattern. And so in this case, the null distribution is basically the difference between the log likelihood that's constrained to fit the other species and the unconstrained log likelihood. So this is better than just looking at the two trees to see that they're the same or different. But it's still sort of problematic because it's using a gene tree in sort of a direct way for phylogeographic research. A more common approach is something that Lacey Knowles developed, which I think of as the coalescent application. And so basically, in this 2001 paper, Lacey conducted parametric simulations within these different models of population and demographic history. And she essentially evolved genealogies that were consistent with these two different models, estimated a statistic from the genealogies, and then compared the distribution of that statistic to her empirical data. And so instead of taking an overtly phylogenetic approach, it was based more on this coalescent approach. And to do this, there's a number of things that one needs to do. And so you need to define the actual demographic model of interest. And so in this case, it's, do these things split into 3 populations or 8 populations? These are different demographic models. Then simulate genealogies under these models and simulate sequence data on the genealogies, estimate the gene trees from the sequence data, compute the summary statistic, decide on the p-value that you want to use, and then compare your empirical estimate to the distribution of values that you get. So it's conceptually easy, but each of these steps is a little bit tedious when you actually sit down and do this. I know because when I was Lacey's postdoc, she made me do this a lot. Maybe, I wanted to do it, but this is an example, from earlier, these 2 models in the grasshoppers. And what we did is we built these null distributions for 2 different genes, CO1 and then this single copy gene. The distributions here are shown for the dual refuge model, so the model on the right. And the empirical values are these arrows, and so we're rejecting these models. The simulated distribution is far greater for the, this is the number of deep coalescences, the statistic that we use. And so the distribution of values is much greater than our actual value. And we use this as a way to interpret or to infer that this history was not a good history for the empirical data. Okay. Any questions about the basics of this before we talk about a few details? So the distributions, there's basically 3 distributions shown here. The dotted line is one, the solid line is the second, and then these bars are the third. And the reason we have 3 distributions is parametric simulations rely on some estimate of parameter values to conduct the simulations. And so in this case, we need to assume a value of theta. And so what we did is we estimated this parameter directly from our data with a 95% lower confidence interval and a 95% upper confidence interval. And we did the simulations 3 times, once using the point estimate of the value, and that's what's shown with these histograms, once using the lower and the upper values. In this case, it didn't seem like the uncertainty in our estimate of theta really made a big difference. All these distributions were outside of this. But I do think it's clear that as the the parameter that we're using to simulate our data changes, this distribution will shift a little bit, right? We also did this for 2 different genes, got similar results, but they weren't exactly the same. And so in this gene, we're much closer to our cutoff value than we were in CO1. And there's reasons why we might expect that this happened. I think there's a better estimate of the genealogy in CO1, so there's some difference there. But in both cases, we sort of looked at this as an independent replicate of the same test. They were single locus replicates or something like this. And so this is certainly a viable approach. It might be a little tedious, but it's perfectly reasonable to do. But I think it's important to remind yourself that what you're doing is you're calculating the probability of the model, and included in that model are the exact estimates of the summary statistics and the parameter values that you're using, right? And so if we're wrong about our estimate of theta, then we're calculating the probability of a model that's not a very good model. And so because we have we're forced to rely on these point estimates of some parameter values, it's you can potentially mislead yourself, and there's a higher false positive rate if you're not doing a good job of estimating these parameters. Does that make sense? And so in this paper, we actually had something in the discussion about whether hypothesis testing is really the way to go. At the time, it was a reasonable thing to do. I've since come to sort of be skeptical that it's the best approach to take for phylogeography. And the reason is that there are all of these assumptions that one has to make in order to believe in the results of the hypothesis test. And so we have to believe that our values of the parameters that we're using are accurate, that our sampling is good, that the model has timing that matters to the organism, et cetera. We also need to, and this is the part that I think is really tricky, the summary statistics that we use can change the results of our tests. And so we use the number of deep coalescences because we thought it had more power than some of the other summary statistics that we tried. With some of the other statistics, we would either get values that were really far from the distribution or closer to the center of it, and we couldn't figure out why. In some cases, they seemed to have one answer. In other cases, they seemed to have other sort of results. And it's difficult to sort of simultaneously assess the adequacy of the summary statistics while you're trying to minimize the type 1 and type 2 statistical error, while you're trying to make sure that things like your variance and the theta estimate isn't influencing the results. Because there's so many moving parts here, and because you're forced to make so many assumptions about the adequacy of things, if you reject a model, is it because the model is actually really bad or because some other aspect of this is wrong? And so I don't know of good ways to address some of these questions. I'm happy to talk with people about that. But one of my frustrations with this approach has always been, well, you do it, you get a result. Do you believe this or not? And there's reasons that one might believe it in some cases and not in other cases. Yeah.
And a lot of your criticisms would also apply to ABC too with this. Like the adequacy of summary stats and things like that.
Certainly. And we're going to talk about ABC here in a little bit. Okay. There's sort of a philosophical issue here too. We reject these models. This implies that one or more of our assumptions are incorrect. But I think conversely, if we fail to reject some model, that doesn't necessarily mean that we've gotten all of these assumptions correct. It could be the case that we're wrong on assumptions that sort of counterbalance each other or cancel each other out in some sense. You just never really know. And these criticisms, I think, are, in general, any model-based approach, regardless of if it's ABC, frequentist hypothesis testing, the Frappel stuff we're going to talk about, you need to think really carefully about the different assumptions and how being wrong in various aspects of it might influence things. And more importantly, and sort of more philosophically, if you do hypothesis testing, you're sort of implicitly assuming that what we want to do for phylogeographic research is try to identify hypotheses that we can fail to reject, right? If we have one or two models, this is relatively straightforward. But what happens if you have several different models and there's two models that are really different, but you can fail to reject both of them? You know, you're sort of left with this situation of it might be model A, it might be model B. You know, by virtue of how you've set up the machinery of the hypothesis testing that you're doing. Other statistical philosophies would argue that when we do research, we should try to assess the absolute probability of these hypotheses. And so rather than trying to find something that we can fail to reject, we should just calculate the probability of this hypothesis or this model directly. And so this would be sort of a traditional Bayesian goal, is just let's assess the probability of the model. And I think the sort of third way that we can think about models and hypotheses is more of a comparison among the relative probabilities of models. And so we could try to look at the evidence that we have and see if the evidence supports one model over a second model, and if so, how much more the evidence supports model 1 than model 2. And these are all sort of reasonable things that might help us think about the fit of our models, but they're sort of philosophically different, and there's different approaches that we might take to it. The top one is usually sort of seen as a goal of frequentist statistics. The middle one is Bayesian. And then the third one sometimes gets called likelihoodism or information theoretic kinds of approaches. And there's shades of gray here among all of these. So I'm dramatically simplifying in cases. So Brian mentioned ABC. We're all familiar with Bayesian techniques. It's, I think, really common for evolutionary genetics and phylogeography. At sort of the basic level, a Bayesian approach to science is we want to assess how probable a given hypothesis is given the data that we've collected and the prior knowledge that we have. And on the surface, this goal, I think, is admirable, and it's something that I can completely get behind. You know, if there are ways to do this, this is a really reasonable thing, right? Because we can make a probability statement about the models that we're evaluating. To do this, usually Bayes' theorem is used, and so there are different formulations of this, but in all formulations, it's a statement of the relationship between the data that you have and the model that you're using to describe the data. They're derived from this conditional probability statement. And so basically, we have this statement of the likelihood, which is shown in red here, the probability of the data given the model. We have the posterior probabilities, which are here, that depend both on the likelihood and the prior probability. So there's this conditional relationship between the prior probability, the likelihood of the model, and the posterior probability that we get out. And so this conditional relationship allows us to make this assessment of how probable these models are given the data. And these are related in a lot of ways to maximum likelihood estimation, but I think there's a couple of differences. So in maximum likelihood estimation, we have some likelihood function. We have different values of some parameters of interest, and we try to find the value that maximizes the function that we have. In some cases, we can do this directly with these sort of analytical solutions. And it's great when this happens and when we have sort of an analytical approach to taking a maximum likelihood estimate. Usually for phylogeography, the parameters and the data that we've collected are complex enough that something like this isn't directly possible, right? And so approaches like ABC are more generally known as likelihood-free methods that allow us to make some assessment of the probability of our data given the model for these complex situations where there's not an analytical solution. And so the reason for this is that we just have too much going on to effectively calculate a likelihood function. And so ABC, and the solution that ABC proposes, is that we can use simulations and approximations to get information about regions of parameter space that are probable given the data that we've collected. Okay, so that was a gross oversimplification, but the point here is that these likelihood-free methods like ABC, you're basically forced to use approximated methods because the analytical solution to the functions that you might be interested in an absolute sense either don't exist or can't be implemented. Is that fair, Laura? Stop me if I say anything blatantly wrong, please. Okay. And so given some prior distribution of parameter values, we can obtain a posterior distribution. And so in the ideal case, you might have a really flat prior, and then your posterior distribution shows that there's a certain region of parameter space that maximizes the probability, the posterior density. So we've all, I think, seen ABC applied to phylogeography. And I put a paper in the shared folder by Fagundes et al. That is a really good example of how one might use ABC to make phylogeographic inferences. And so what system did this paper investigate?
People.
Which one? Humans, right? And so human evolution is something that, of course, lots of people are interested in, and that many, probably hundreds of papers have been written about the demographic history of humans. And so what Fagundes et al. Did is they basically summarized some of the primary theories of human evolution into these three different classes of models. There were these multi-regional models where humans evolved both in Africa and Asia and other parts sort of in parallel. There were these out-of-Africa models that were either with secondary contact with Homo erectus, with our immediate ancestors, or without that sort of secondary contact. And so fairly complex demographic models, right? The models include potentially things like gene flow, changes in population size, different branching events between Africa, Asia, and the Americas, etc. They have a nice data set that they use. And in the paper, they point out, we can't analytically compute the probability of these models, but what we can do is we can use these approximate Bayesian techniques to calculate the relative posterior probability of the models, right? And when they do this, so this figure shows 8 models. The approach that they take is a pretty reasonable one. They say that, traditionally in human genetics, there have been kind of 3 classes of models, which I just went through. And so what they're going to do is they're going to calculate the relative probability within each class first to figure out what model is the best model for each of these 3 broad kinds of scenarios. And then they're going to compare secondarily the best model in each class to each other. And they don't state it directly, but they sort of imply that the reason they're doing this is that they will get better results when they have a smaller number of models in the comparison. And so for ABC, why is that the case? It's related to the prior probability, right? If you have 2 models, what's the prior probability of either of those models? 0.5. And if you have 8, it's 1 divided by 8, right? And so as you add models, the prior probability decreases. And so one of the reasons that ABC in practice tends to work more effectively with a smaller number of models is just those models have a higher prior probability to start with. So they go through and they do this, and the models with the boxes are the ones that have the highest posterior probability in these 3 individual comparisons. And then when they compare them to each other, they find that this out-of-Africa model has a posterior probability of nearly 0.8. And so it's well supported. Yes?
What do you mean by effectively? Like ABC models work more, like, what's your metric for effective run?
What context?
Well, so you said like ABC models work most effectively if you have small number of models. What do you mean by effectively? So you mean like
if you measure something like the Bayes factor between the model with the highest posterior probability and the next highest model, that Bayes factor tends to decrease as you add more and more models to the comparison.
So most definitive comparison between models is effective.
And that makes sense, right? If there's 100 models, for example, the models are very similar to one another potentially, and it's harder to discriminate among those, right? And so to get nice definitive results with ABC, you would compare models that are different from one another, and your data would favor one strongly over the other. So how many people work in empirical systems that have benefited from hundreds of published papers, previous research talking about models? None of us, right? So one of the things that when you talk to people who do ABC, or you talk to Nelson Fagundes, they'll say, well, you just have to pick models that are reasonable and that people think are good models, and then you compare them and you can calculate these probabilities. And so this is a reasonable approach for human geneticists. It's a little trickier to apply if you work in some of the organisms that we work in this room, right? And so one potential complication with ABC is just developing the models to compare in the first place. Okay, so I wanted to put this up here just so that people had an understanding. How many people have actually sat down and done an ABC analysis? So most of us. So I think we all have the basic understanding here. Have you used the R package, like Sillery's R package? Is that something, have people written their own scripts to do it? Some people have probably done that. So there's a number of ways that you can do it. All of them pretty much start with replacing the data with some vector of summary statistics, and then you simulate and you calculate this vector from the parameters in the prior. You define some threshold, and your likelihood is proportional to how close your simulated data are to this target in your empirical data. Usually if you use multiple models, you'll use some sort of rejection algorithm to identify the relative support for model A versus model B. Something like that. So it's a very nice approach. It's really flexible, and so you can compare models of any amount of complexity that you want. And I think this is a really positive aspect of it. You can also, of course, estimate the parameters given the model. And this is a really nice thing about it. When you do this, the summary statistics matter. And sometimes your summary statistics will do a better or a worse job of estimating different parameters, right? So for people that have done ABC, how did you decide to use the summary statistics that you ended up using to summarize your data? Yeah.
I have used simulated different summary statistics, and depending on if they show different results for each scenario, I pick those because they are actually showing different things instead of simulating the same thing.
So you did some exploratory analysis first to try to find a statistic that was unbiased or something like that.
Or that will produce different results depending on the model that I'm creating.
Right. So if you know that a summary statistic, when you simulate the data under model A, is likely to identify model A as the best model, but if you use the same statistic with model B and then it identifies model B as the best model, that's relatively unbiased and you're not sort of influencing the end result that you get. And so this is some kind of approach like that is recommended. There are a number of theoretical papers that have been written about different kinds of statistics and different kinds of approaches to using them. Some people have criticized ABC because it's hard to demonstrate absolutely that any given statistic is adequate. And if you use inadequate statistics, then you can bias your results in subtle ways. So there's, to break it down into 4 steps, of course, there's more going on than that, but that's a general outline. So one thing about ABC that's, I think, also useful is that when you estimate parameters, you have a really good understanding of the shape of the distribution of these parameter estimates and how much confidence or credibility you should place in them, et cetera. And so this is a figure that I borrowed from Mike Hickerson. When Mike explains ABC, he says it's really easy. You just follow the flow chart, and you get the answer. But to break down the different steps in the typical run a little more directly. And I put this up only to make the point that, like parametric bootstrapping, you make a number of choices along the way, and it's possible that these choices will influence the results in subtle ways. And so the more careful you are and the more you sort of explore each step along the way, the better your results are likely to be. And so it's interesting to, I think, read the Fagundes paper thinking about models and how you model the data. And so what they basically say is that they model these 3 different scenarios, and there's some uncertainty about the details, so they use these different growth models, et cetera. And it's unclear, even after this analysis, exactly which of these is the best descriptor. They imply that they can do sort of further analyses once they have identified this model as being the best. You might estimate more statistics or more parameters from that model sort of secondarily. So we've done a little bit. So this is a paper that my grad student Tara Pelletier has in press right now. We were, and have been for a while, this is a paper took an embarrassingly long time for us to write. And so we actually started working on it while we were at LSU still, and it's only now accepted. But we wanted to apply ABC to this system of salamanders from the Pacific Northwest where there hadn't been much previous work done. And the previous work that had been done either assumed or argued in favor of one of these 5 models. And so what Tara, like a lot of graduate students, sort of her subtle goal was to prove me wrong in some ways, because at some point in a paper I had said that this model of just strict isolation was probably the best way to think about this system. And we did an ABC run, she did an ABC run, And we found that indeed that model had the highest posterior probability. So it was 0.7. If we look and sort of evaluate how strong of support that is, the Bayes factor between this model and this model is about 4.7. And so in sort of a typical way of interpreting Bayes factor, this is seen as moderate support. And we could estimate parameters that seem believable using that model. And so we should have stopped here and published the paper. But what Tara was curious about, and what we started talking about, is how do we know that this model is really the best model, particularly when we know we have a system that is heavily influenced by post-glacial expansion, because half of the range of the species was under ice about 20,000 years ago. And so it seemed to us like expansion might matter. But the one model that people had proposed in the past that had expansion in it had low probability, possibly because it was a model where everything was in the same refuge during the Pleistocene. So it wasn't like an isolation with expansion model, it was just a strict expansion model. And what Tara was really curious about is how the probability of this model that was the best in the first comparison, how relative was that probability? Is it the case that that's a generally good model and most of the models we compare to it would have a lower posterior probability? Or did we just do a bad job at picking models to compare and this was really the best of a bunch of really bad models? And so we know when we do ABC that we calculate relative posterior probabilities, and the question is just how much does that matter? Broadly, this is a question about the models that we choose to include in an ABC analysis. Because generally a small number of models are used, we have to be selective about choosing these models, right? And so what Tara did is she defined model space such that we had about 140 different models that were either a single panmictic population or two diverging populations. Some of the models had gene flow, some had population expansion, and so when you put all these combinations together, we had approximately 140 different models that we could choose from. Using this model 1000 as a starting point, she picked 4 of these. I keep saying we, like I actually did it. She did all the work, right? She picked 4 other models at random and repeated this analysis. And basically, we wanted to see how the posterior probability of model 1000 changed as a function of the other models that we included. And so, is model 1000 a good fit in a non-relative sense? We found that the mean posterior probability of this model was about 0.44 across 100 replicates. It ranged from 0.21 to 0.77. And so one way to think about this is that we got really lucky with this initial comparison because these other models were so bad that the posterior probability of this model was relatively high in comparison. Don't do this at home. I mean, it took a lot of work to sort of get to this point, and then we didn't have any good way of sort of interpreting those results, right? And so to visualize this, if you do an ABC analysis with all of these models, and I think this might be a record for the most models in an ABC analysis, here's where model 1000 ends up. This is, the models are on the x-axis, they're just ranked by their posterior probability, and so that's on the y-axis here. The red line is the prior probability of each model. And so some of these models are above the prior probability, but none of them have a really high posterior probability. The blue line is our best guess as the highest posterior probability that you would expect a false model to have it by chance. And so we did a lot of other simulation testing comparing how the number of models influenced our ability to choose the best model and doing some randomizations. And what we basically thought is that any model above the blue line was a good enough model that we might sort of use it for further analyses. We also did some posterior predictive simulations to try to calculate the absolute fit of these models. And unfortunately, we found that we didn't have a really strong correlation between the absolute fit measured by posterior predictive simulation and the relative posterior probability. So we're not quite sure what that means. There's a couple of interpretations about that. So I'm happy to talk more about this, but my main point here is that in an ABC analysis, your choice of models matters. And it's hard to know which models to choose if you're working in non-model systems. Okay. And I think this is in some ways justification for an approach like FRAPL, where you define models not sort of based on what you think you know, but just based on some pattern in parameter space. And we'll talk more about this. And so, but just to summarize this ABC approach. I think the goal of like a Bayesian statistical analysis is a really good one. We want to know how probable models are given our data, right? But at least for phylogeography, this is difficult to calculate, and we're forced to use things like these approximate methods that calculate the relative posterior probabilities. And we need to be careful with that at a minimum. And so I think these analyses can be really useful, but you shouldn't misinterpret, and it's really tempting when you get these posterior probabilities to think this is a good model. It's only good compared to the other models that you have in your set. All right, so yesterday I was talking a little bit about ways that we might try to use model selection as an approach to doing phylogeography and making phylogeographic inferences. And we talked some about parametric bootstrapping and null hypothesis testing and ABC. This characterization here that you can think about the way we assess evidence in biology as being either in a frequentist approach, a Bayesian approach, or a likelihoodist approach comes from this Elliott-Sober book, which is a kind of sort of a philosophy of science kind of book. But it's actually, parts of it are really interesting and worth looking at. I don't know if you've checked this out, but it might be something that you're interested in. And what Sober basically argues is that in a practical sense, what biologists want to do is they want to evaluate hypotheses by figuring out what kind of evidence they have and how the evidence favors one hypothesis over another. And particularly, Sober makes the distinction between the kinds of experimental research that some people in the biological sciences do and then the kinds of historically motivated questions that many evolutionary biologists use. And to Sober, we shouldn't necessarily think about historical sciences in the same way that we think about experimental kinds of sciences. And I think those of us in this room, this makes good sense because what we do isn't experimental in the classic sense in any way. We don't have replicates, we're not setting up trials, we're in field plots and these kinds of things. And so just again to sort of review, we've been talking about model-based inference, and one of the reasons we want to use models for these kinds of historical sciences is that essentially they allow us to get signal and infer process from signal in the data that has a lot of stochastic variation within it. And certainly genetic data we know are full of various sources of stochastic variation. The two important ones are the coalescent process and the mutational process, which are essentially superimposed on any of the sequence data that we collect. And so the reason we want to model our data is to make inferences without being sort of misled by these various sources of stochasticity. It's, I think, important, and I like this quote by Soule, that basically encourages us to think about models as tools that we're using to make our thinking and our thought process better and more strict, rather than something, a black box that we just pour our data into and we get an answer out of the other end. I'm sure that the discussions we've had about Frapple have impressed on everyone that we don't want you to use this like a black box, and that, particularly when it's in development as it is, and probably will be for the next however many years, we need to really think about what's going on and do that. One of the motivations for this workshop is to improve the software, of course, but also to get a group of people who are really familiar with it so that when you guys write papers and you hopefully use Frapple, other people will read these and they'll see how creative people have been with applying it in different kinds of questions. And so models then allow us to integrate basically all of the things that we're interested in and that are going to influence our ability to make inferences. These hypotheses, the data, the assumptions that we have for the statistical methods, estimates of the model parameters, and also we can do things like estimate covariance among parameters. This is probably really important for things like divergence time and theta, where these will covary to some degree. Okay. Now what's interesting to me is that the ideas that Sober outlines in this book and that other people have talked about are actually particularly old ideas, and they predate experimental science of the kind that we sort of classically think of. By a pretty wide margin. And so Thomas Chamberlain was a geologist who was working in the 19th century, basically. And geology, of course, is very historical in nature, the kind of science that geologists do. And what Chamberlain's research basically was designed to do was to evaluate a small number of models that represented these historical hypotheses. And Chamberlain approached things by deriving a small number of plausible hypotheses and then trying to evaluate the empirical data in the context of these hypotheses. And so if the data was fossils from a certain strata of rock, did this favor one hypothesis over the other? And so the spirit of what I think we should be doing for phylogeography is consistent with these ideas about how one should conduct this entirely different kind of research in a different field of science. Okay. And so it's, I think, also worth thinking about the relationship between our models or our hypotheses and the observed data in different frameworks for analysis. And so in null hypothesis testing, we're calculating essentially the probability that our observed data is greater than or equal to some expectation under a null hypothesis. And so the data is conditioned on the hypothesis, in essence. It's reversed if we seek to evaluate multiple models in the way that sort of Chamberlain envisioned, because we're actually calculating the probability of individual hypotheses conditional on the data. And so there's a subtle switch in how we're doing things, but it allows us to do the kind of multimodal inference that we have talked about, where maybe the real answer isn't any of the models that we're including in the set, and we're sort of integrating the uncertainty in that by estimating parameters under each model, comparing the parameter estimates. Particularly if you're doing comparative studies where divergence time is important, this kind of multimodality might help prevent you from making errors in the estimates of these parameters that you're actually using to make the inferences you want to make. And so it's possible because we're switching these relationships around. Okay. And then the other thing that is, I think, important to think about in the general sense is that even if we have a large number of models, so many more than the 4 that Chamberlain would have used, for example, it's probably not the case that the truth is somehow one of these 1,500 models that's in the migration array for the 4 population or something. It's reasonable to suspect that all models are inadequate descriptors of the past. The past is really complicated. Our data are probably not a perfect sample from populations. There's all sorts of reasons why these models are likely all wrong. And George Box, this famous statistician, is largely famous for this quote, basically reminding people that even if models are wrong, they're still likely to be useful in some ways. As an aside, there was a memoir published relatively recently, and it's a surprisingly interesting book. And so the story is that he essentially was forced into becoming a statistician because he was working in a field office during World War II and expected to analyze this messy data, and there were no statisticians available. And they said, George, you're good with math, learn how to do this, basically. And so it's a interesting story, I think, about how practical he was and how many of sort of the ideas of statistics, of course, have developed for really practical reasons. Okay, so what is a useful model? I think a useful model is something that has the right balance between the right amount of parameterization so that we minimize bias and that we don't have too much variance that results from overfitting of the model. And so what we mean by this is basically in a multivariate way, easiest to think about when we think about a simple sort of regression. So we have some number of data points, and we can draw a model of arbitrary complexity to summarize these data. Right? The simplest model, of course, is a line, and the line has some good properties and some bad properties. Right? We could also draw some really complex model that would fit our data better than a simple line. But why wouldn't we do this when we did a regression?
No, you're just soaking up residual error. You're just adding parameters.
Okay. We're soaking up the residual error, and what can't we do with this curvy line that we might be able to do with the straight line? we have limited predictive ability, right? If we have a new point here, well, then our super curvy line doesn't have any sort of fit to that, whereas our straight line might. Okay. So how is this relevant to phylogeography, or is it at all relevant? This sort of balance here between bias and variance. So presumably a model that had a very large, too many parameters might be reasonable for the data that we had, but it might make it difficult to apply that model to either replicate data sets or additional data that we collected. And so the argument against overfitting models in part is your observation is a small observation compared to the universe that you might eventually be able to collect. And you don't essentially want to get carried away and sort of convince yourself that the answer lies in one direction when maybe it lies in some other direction. This, I think, for us is particularly relevant as our field is moving from the tedious work of collecting data on a locus by locus basis to the enticing but kind of scary world of we're going to collect thousands of loci at once. So the types of data that we're collecting is really different. How many people, when they've gone from single or a few locus studies to these next-gen studies, have dramatically cut down their sampling? Most of us, right? So we don't typically see these data sets like Teresa was talking about where you have 400 samples for RAD-seq data, right? And that's sort of an economic trade-off that we're making. Our data are really different. And if we think about the predictability, it's not necessarily the case that 1,000 RAD-seq loci from 20 individuals is a much better data set than the 4 loci, 400 individuals for all kinds of questions, right? We just don't know. And thinking about model fit and model complexity is going to be an important part of evaluating the strengths and weaknesses of the newer kinds of data that we're collecting. Okay. I'm not pretending that I know exactly how to do this, but I think it's something that we should all be thinking about and at least trying to do. Because this is an important, very topical thing that is going to be a big influence on our field in the next 5 or 10 years, I would imagine. Okay, so in general, we want models to be useful. We want them to have a relatively good fit without being so over-parameterized that they lose any predictive ability that we want them to have. And so I think it's reasonable to think of model complexity as being related to how much data we have, and model selection as being a way to select the model that's the best fit for a particular amount of data. In ecology, they've thought a lot about these kinds of issues, the relationship between how much data you have and how complex your model can be. And the typical, if you talk to a field ecologist, an experimental ecologist, they usually say things like, well, you can estimate n over 10 parameters, where n is the number of individual samples that you're taking. I don't think we have a rule of thumb like this for phylogeography, but as I mentioned in the morning, one thing that we would like for FRAPL at some point is recommendations about how much data, how much information your data has, and how much parameterization you might be able to get away with on average. And so that's something that I hope we'll be able to work on. Okay, so I'm going to take a short segue and talk just in general about information theory and about these information theoretic approaches to doing model selection, and then finish up with a couple of examples that we have done on empirical systems in our lab, just as a way to illustrate how we've used this sort of approach for phylogeography. And so we can think about information as something that decreases our uncertainty about the empirical systems that we have, right? When we start a phylogeographic project, we might not know anything other than the purported range of the species that we're interested in. And we go out and we collect samples, we collect data from these samples, we're gathering information, and we can think about the amount of information we gather in a bunch of ways, but one way is it decreases our uncertainty about the things we don't know to some extent. And maybe a good argument for stopping data collection and finishing your dissertation is when the new data that's coming in is no longer changing your mind. It's no longer decreasing the uncertainty that you have. It stands to reason that you get to a point where you stop changing your mind, you stop learning new things, and that might be the time to graduate. teasing Ed. So, yeah.
There's a distinction though between data and information, right? So in terms of like information theory, I mean, information is based on your data, but also your model, right? It's not just the data.
Sure.
So.
And yes, it's probably not the case that you'll reach this point at the same time for every conceivable analysis. I'm speaking in really general terms. And as I mentioned, I don't think we have really good ways of quantifying data informativeness for phylogeography, but I think that's a reasonable goal to have to try to think about this. So information in this context is a construct of statistics in sort of a very real way. And it's easy to imagine that we can quantify information, but it's hard to do in practice. In some ways, this kind of construct is analogous to something like effective population size. It's easy to imagine an effective population size in the abstract, but this is a hard thing to actually count. You know, you can't go out into a field and say, okay, of these 2 million organisms, 100,000 of them are effectively important. You can do this in the retrospect, because it might be only that 100,000 leave descendant alleles X number of years into the future, but it's an abstract concept. And so I think when I started reading information theory stuff, it wasn't until I sort of accepted this idea of information in the abstract that I started to see the utility of it. And so a really good source to start off is this Burnham and Anderson book from 2004. And basically, they talk about information theoretic sources or kinds of approaches in the context of frequentist hypothesis testing on the one hand and Bayesian approaches on the other. And basically what Burnham and Anderson argue is that they're simpler and more applicable than hypothesis testing for many kinds of science, and I would argue phylogeography fits into this, and more easily applied than Bayesian approaches. They require fewer shortcuts in some ways than a Bayesian approach. Okay. So what I want to do then really quickly is just develop a little bit of information and background about how information can be quantified. And this will lead us to AIC and to model selection. And so assume we have a set of hypotheses. This could be 4 hypotheses that people have proposed in the literature. This could be the 78 models that Frappel has in the model array. For each hypothesis, we have some sort of a mathematical model that represents the hypotheses that we have. And with this model, we can estimate model parameters under a maximum likelihood framework so that we can get an estimate of the maximum likelihood of the function represented by the model. So we get a maximum likelihood estimate given a set of parameters for these models. Okay? So these are what we're doing with Frappel, basically. Once we have that, we can start thinking about how to compare models and make the model comparisons based on a couple of pieces of information. The first one we need is this abstract idea from the statistical literature by these statisticians Kalbeck and Liebler. So in 1951, they wrote a paper that basically argued that we can think of information and the units of information as the distance between full reality, which we can't ever really know, and a model that describes full reality. And the degree to which a model approaches full reality is the information distance between our model and our reality. This is all abstract, right? And so basically they said there's some, potentially if we knew everything about full reality, we could describe it, and then we could measure the distance between that full reality and the model. So it's basically information is what we lose when we use a model to describe reality. It's sort of the opposite way of looking at that. And so what these information theoretic approaches seek to do is identify the model that has the minimal amount of information loss between full reality and that model. So of the models we're thinking about, which one gets us closest to full reality? And in their paper, they said it would be really nice if we could measure this in reality, these abstract concepts, but it's hard to do. And it's hard to do because we don't know what full reality is. And there's one of the few sentences in this paper where I can read and understand completely exactly what they're saying. They say something to the effect of, it would be nice if we could quantify reality fully. After many years of thought, we haven't been able to And so reality is complicated. The reason we have models in the first place is we want to make some sense out of it. And so they define information using this sort of integral equation approach in the theory, and say for most systems we can't hope to ever calculate this because we don't have knowledge of full reality. It's certainly true in genetics. And this is kind of where this paper ends. About 20 years later, Ake came up with a way to link this theoretical construct that Kolbeck and Liebler came up with to standard statistical theory. And basically what he found is that if you estimate parameters under a model, you can come up with an expectation on the model of full reality, even if you don't know what it is. And he did this by decomposing this expectation in a way that made it equivalent to the probability of the model when it was conditioned on a particular data set. And so he came up with this double expectation here. And the entity is related to the maximized log likelihood function for a model, but it has a certain amount of upward bias to it. And what Ake found, and this is where what he did became really important, is that the amount of bias that these kinds of models have is roughly proportional to the parameterization of the models. And so Ake in this paper suggested correcting for the bias by penalizing models that were more complex when you did these comparisons of models. And so this doesn't probably look familiar to most of you, but you can reduce that equation to this, which is something that I think we've all seen and we've all used. And so this is the classic AIC formula, where -2 times the log of the likelihood of the model given the data, plus this penalization term for the parameterization of the model is equal to the AIC. And so you can think of this as this deviance term and then a correction for model complexity. Okay. And so the reason that everything is multiplied by -2 is because this was sort of a known entity in statistics, this deviance term. And Ake rephrased this to make it more palatable to his peers who were used to thinking about deviance in statistics in this way. So basically, what AIC is measuring is the difference between full reality, which we don't know, and the models that are being compared in the model set. Okay? So we can think of the deviance term as a measure of how poorly the model fits the data. It's reasonable then that as we increase the number of parameters, we would expect this term to decrease in its magnitude. And we're trying to find the balance between parameterization and the deviance in these models. So what we don't want to do basically is get to the point where we're modeling the noise, just like with the Kirby line and the regression. And so this AIC equation is allowing us to penalize really complex models that don't add any additional information about reality to the system. Okay, so we have a trade-off between variance and bias. And it's interesting, I think, to think about, but this is really related to this idea of parsimony in a general sense, right? Good models have this ability to be fairly simple and to make predictions outside of the sample size. And so this is related, of course, to this idea that simple explanations, Occam's razor, simple explanations are to be preferred if there's no good reason to prefer the complex one. Okay. One thing that's there's a couple of things about AIC in general that are important to recognize and that sometimes you'll hear and see people not get these points. So the most important one is that there is nothing about the absolute values of AIC that matter. So one AIC score for one model doesn't tell you anything about anything. It's only when you compare them and compare the differences between models that you can measure the amount of information that you're losing by doing the model comparisons. Another thing that's important to notice is that there are corrections for sample size. And so even though the sort of normal way that you see the AIC equation written is at the top, there's this bias correction for small sample sizes that is usually introduced. And so for Frapple, I think we don't do that yet.
Right. And we don't know how to measure n.
We talked about that. We don't know how to measure n. So one of the things related to this idea of quantifying information in a data set is we want to be able to correct for small samples, and presumably, hopefully, someone will figure out the good way to measure information. So we've talked about, is it number of samples, is it number of loci, is it the product of these two? We've kind of gone around and around without really getting anywhere about that.
And also good to notice, I mean that ratio, the correction factor, n over n minus k minus 1. So when n gets big, n versus n minus k are pretty much the same value, right? So it converges, but I think Berman Anderson say like when n is 40 times k, don't bother, it doesn't help anything.
But I think for many of our data sets, we're not to that point yet.
Unless we measure the number of samples or
So we should probably think about correcting for this at some point. Okay, add it to Nathan's to-do list. It's so much fun to have a postdoc. Okay, so another thing that's, I think, worth emphasizing is that there's no requirement for the models to be nested for these information theoretic approaches. It's not like a likelihood ratio test or a Nyman-Pearson test where you have to have a nested model. A second thing is that if the difference in AIC between two models is about 2 units, and the models have one parameter, so one model has one more parameter than the other, you need to remember that these are essentially equivalent, because the only difference in AIC score is caused by this penalization for the extra parameter. And so this is typically called a pretending variable. And another sort of rule of thumb that people who do this in general pay attention to is that if R is the number of models and N is your sample size, if your sample size is smaller than the number of models, then you should think about your data analysis as exploratory. And so, for these 4 population models where we have 14,000 migration arrays, most of us probably don't have data sets, however we measure them, that will be bigger than that. Right? And so there are statistical sort of reasons to maybe be cautious at that point. Now, again, it depends on how we measure information. Okay, so how do we use AIC scores for model comparison? The first thing we need to do is to define basically just the difference in AIC scores between two models. So we can calculate these AIC differences, which are usually abbreviated delta I, as just the distance between an individual model and the best model in our comparison. And so models that are close together will have small delta I's. If a model is extremely bad and is really far from the best model, the delta I will be big. And so the cool thing about AIC differences is, in theory, they represent the expected Kullback-Leibler distance between the best model and the model that we're considering, the ith model. These delta I's are additive as long as the scale that we're using is the scale of information. Again, this abstract statistical concept. And so the easiest thing to do in a model comparison with information theory is just rank models by the delta I differences. I think, though, it's really useful to quantify the total support that we have in our data for the set of models. And we can transform these delta I's into something that's it's directly analogous to relative posterior probabilities. And these are called model likelihoods. And so because when we estimate parameters, we're estimating the parameters of our data given these models on the likelihood scale, we can approximate this by taking an exponential of half of the differences between these delta I's. And so what this does is it basically allows us to transfer the AIC differences into a relative likelihood of models in a way that allows us to make model comparisons. So if you take, and Frappel will do this for you, the exponential of -half of these delta I values, you can then add up all of these measures for all of the models and divide an individual model by the total in such a way that you get the relative proportion, relative probability of an individual model. Okay, so you do that in this way. So these are called model likelihoods, abbreviated WI, and you take an individual model, which is shown in the numerator here, and divide it by the sum across all models, the denominator. This gives you a relative probability. If your model is much, much better than any other model in the set, the numerator and the denominator will be very similar, and your value will approach 1. So these essentially represent the probability that that model in the set is the best model of the set. So if you see WI values of 0.9 or 0.95, then there's a high probability that that individual model is the best model in the set. It doesn't mean that that's the best model or the truth in any way. It could be that there are other models that are not being considered that if you put them into the set would dramatically change those. But it does give you a way to evaluate of the models in the set, which is the best one.
And
It's a measure of the confidence you would have in that.
these are often called KK weights or AIC weights. You may have seen that instead.
So. And again, they're equivalent to, and they can be interpreted in the same way that you interpret Bayesian posterior probabilities. So I think a really cool thing to do, and something that I'm sure we'll do in one of our Frappel papers, is to do an ABC analysis and an information theoretic analysis with the same sets of models and the same data. I would hope that we would get very similar results. But it'll be interesting to see if that happens. Okay. Another really good source of information that's very readable is this 2008 book by David Anderson. It's called, what is it called here? Model-Based Inference in the Life Sciences. And it's very non-mathematical, very readable, very short. And so, but what Anderson argues basically is that science should be about evaluating models and comparing how good one model is to some other number of models in the set. Anderson comes down on the side of the debate where he wants you to choose a small number of models. So in our case, it would be a priori hypotheses and evaluate 5 or so in the model set. He would probably make this argument largely because then you have many more data points than models, and there's more statistical validity to doing this. Once you evaluate these models, though, what Anderson does is he says all of our inferences should be made using parameter estimates from multimodal inference. So we've talked about this, but I just wanted to put an equation up. This is essentially model averaging, where, for example, if we have divergence time estimated with 3 different models, we might get 3 slightly different estimates from each model. We would take the model probability of an individual model, multiply the divergence time by that model probability, and then sum those across the 3 models to get a model averaged estimate of divergence time. And so this is a good way, I think, particularly for comparative phylogeography, because if different species have different models that are optimal, and they still include a common parameter, you can then directly compare those parameters without the uncertainty that comes from having different models. Any questions about model averaging? Okay. All right, let's talk about biology then. And a couple of data sets from my research group where we've done this. The first one I'm going to talk about is Salix melanopsis. This is a willow that is found on riverbanks. And these samples date back to my dissertation. And in some ways, I'm still working on finishing my dissertation, I guess. But what basically we have is a bunch of willows that are collected from river drainages in the Pacific Northwest. And so we've been interested about understanding how these samples responded to glaciation. The reason for that is that this dotted white line here is the maximum extent of glaciers in this region. It happened about 22,000 years ago. And so many of the organisms that we can collect are occupying habitat to the north of this glacial line. So clearly they haven't been there too long. We want to know about Pleistocene population dynamics. Okay. So what we did really quickly for this paper is I actually, before I left LSU, I had some startup money that I wanted to spend before I moved. And so we designed capture probes for the species and then sequenced about 40 individuals on a Illumina GA2X. So this was several years ago. And we ended up with a bunch of data. We did some assembly and whatnot. And ended up with what we thought were about 414 loci that represented good data that were not paralogous that we had some confidence in. And one of the really surprising things, and the interesting thing to me about this, was that prior to this point, we almost never worried about loci that didn't have any variation because we were always screening. And only sequencing markers that we found SNPs in, right? And so about 3 out of 4 of these loci were invariant. And so this gives us a much better estimate of genomic diversity than just looking at loci where we do screening. We had done 5 loci previously where we had screened these loci from a genomic library, and they're shown with these red bars in terms of the variability of these loci. And so I think we clearly had a non-representative biased sample as a result of the screening. We also clearly have some loci that are huge outliers for the number of SNPs. I think this is SNPs per kilobase. And we think, and we didn't actually end up analyzing these, but we think that those are likely paralogous loci because they have so many more SNPs than we would expect. However, we didn't have a really good way of deciding where to draw that line for paralogous loci. We happened to take these out just because there was a little gap, but it's probably just a really stupid reason, who knows. But we just didn't entirely trust those markers. The fact that we had other loci from Sanger sequencing that we didn't think were paralogous, there was nothing to indicate that they were, and they were sort of overlapping with some of these, made us think that maybe those were okay. Most of our loci, though, had 1 SNP or a very small number of SNPs. The other cool thing about these data is because I collected them, they weren't really attached to anyone's project, and so I used them for a class that I taught at LSU, and actually we analyzed the data and wrote the paper as sort of a class project. And so the grad students who helped me are all shown here, and they're all co-authors on this paper. So what did we do? We did a bunch of things because I had like 10 or 12 people to do analyses. So I was just like, do this, do that, do this. It was awesome. But one of the things that I really liked, a guy named Brad helped with, and you can use migrate in a model selection framework. And so Peter Beerley has really, he's been working on migrate for well over 10 years, and it just gets better and better all the time. And recently he implemented this model selection framework that uses, it's in a Bayesian statistical framework, but it's model selection in a directly analogous way to information theoretic framework that we just talked about. And basically what you can do is, of course, you can estimate a full migration model in migrate, right? That's what it does. It's an island model. And we did this between these 4 populations, the salmon drainages down here, the Clearwater drainages, the northern drainages, and then the western or coastal drainages. And for 4 populations, the full migration model would have 8 migration parameters and 4 theta parameters in it. What we can also do, though, is we can estimate different submodels that represent specific or particular post-glacial colonization scenarios. So for example, maybe we think in model 1 that these 2 regions acted as refugia and they supplied colonists to the other regions. We can model that using this and a number of other ways. And so we decided to take this model selection approach, model a bunch of different demographic models using migrate, and then see which one was the best. The data we used for this were the loci that were variable. And what we found was really interesting, and I think it represents a really good result. So this model, where there were functionally 2 different refugial areas in the Clearwater and the salmon, was supported as best. But interestingly, we found that if we modeled migration from only the Clearwater to the coastal populations, and only the salmon population, which is southern to the northern drainages, that had a really high model support in this framework. So 0.99976. And this seemed curious initially that there wouldn't be any gene flow between these contiguous regions in the Clearwater and the northern drainages, but apparently in other systems there's been indication that there's a barrier between these 2. And the suggestion is that the salmon was connected along the, sort of along the ridge of down at the eastern ridge of the Rocky Mountains to some of these northern drainages, and they kind of went around and over somehow. And so there is some evidence for this in the literature for the biogeography of this region. Of course, this is somewhat limited because the models that we're using are all island models that don't include population expansion. And I'm sure all of you are sitting there thinking that this is bull because I told you that this was a post-glacial system. And it's reasonable to think that population expansion matters, right? So we also did information theoretic approaches using a different program that included expansion. And this program, actually I wanted to make one other point before I get there, so hold on to that. This was really interesting about why we don't want to use parameter estimates under full models to make phylogeographic inferences. So here's a table of parameter values estimated under the complete migration matrix. And I've just highlighted the migration estimates between the Clearwater and the northern drainages. And so remember, this model has almost all, there's almost no support in model space for any sort of gene flow between these two regions. But if you look at the parameter estimates, 3.45, 3.6, they're comparable to many of these other values. And you could easily look at this and assume that migration between these regions was important if you were just judging this on the basis of parameter estimation. In a similar way, if you looked at gene flow between the northern drainages and the coastal, these values are fairly high too. So as soon as you have parameters in a model, any program is going to distribute the genomic variation in such a way to sort of optimize the overall likelihood. And that's going to lead to different patterns of parameter estimation than you might see under a different model. I think clearly what we want to do is select models first, and then only subsequent to that worry about parameter estimates. But as a field, what we've been doing is sort of skipping this middle part, or doing the model selection just in a qualitative way. Well, I think I should use migrate, I think I should use IM, whatever. And then trying to make all of our inferences on parameter estimates. We're making it much harder than it needs to be by doing it in this way. And certainly one of the main motivations that Brian and I had for Frapple when we first started talking about it was to provide people with a tool where you could figure out which model is best to use to do the parameter estimation. We think that it's entirely likely that Frapple will never be as good at estimating parameters as some of these other programs. But at least I think it's, there's reason to think that. But if it gives you a tool to say you need to use IM to estimate this parameter, then that's a huge help. Okay, so let's move on to the second way we did model selection in the system. We used this program DADI that Ryan Gutenkunst et al. Developed. And this uses diffusion theory and allele frequency spectra to calculate the composite likelihood of a variety of models given the data. So like Frapple, anything that you can imagine in MS can be put in as a model to DADI, as long as you have no more than 3 populations. And the reason for this is apparently it's very hard to calculate allele frequency spectra from more than 3 populations and to do the diffusion calculations that are required to make this work. So you summarize your empirical data using allele frequency spectra. You calculate the expectation of the allele frequency spectra given your demographic model and your parameters using this kind of math called diffusion approximation. And then you can calculate the probability of your data given the model by composite likelihood. DADI is a really good program to use if you have SNP data, because your allele frequency spectra is always, the empirical data are summarized in the same way regardless of whether you have 100 loci or 100,000 loci. And so runtime is not necessarily a function of data size in the same way that it would be for programs that are Markov chains or something like this. So we parameterized a number of models, but we had this constraint about only looking at 3 populations. So for this analysis, we restricted ourselves just to the inland samples. So the salmon, the Clearwater, and the northern drainages, and we ignored all of the coastal ones just because we wanted to leave out, we had to leave out 1 population, basically. So we can make a really complicated model, and we can calculate the log likelihood of this model using these composite likelihood approaches. This doesn't get us anywhere on itself, but if we have a large number of models, then we can choose the best model from the set. So how we chose models to include, there's many models for 3 populations that include migration, expansion, population size change, and all these things that we could have imagined. So we went through the literature and we summarized different models that people had proposed in various places. We also, the class came up with some as well. And so like if you see something like the Brunsfeld model, that was a model that Brunsfeld had proposed in some paper. And so we ended up with 12 models that we did the study analysis for. And we calculated the likelihood AIC values and model probabilities. And it turns out that this model had the highest model probability, which surprised me because there was no expansion parameterized in the model. And I had always assumed that this would be the case, that we would need to parameterize expansion. Other analyses with smaller data sets of ABC analyses have indicated that we should, in fact, be parameterizing expansion. And so there's something fishy about this in some ways. But again, we had many models that included expansion in our data set, and plenty of opportunity for those models to have a high probability of the data given the model. So there's a couple of things that might be going on here. One would be that all of our models here are modeling basically divergence from these three populations, right? And so it could be that by doing that, somehow the signature for expansion is degraded to the point where it doesn't make the probability of the data given these models very high. That's, I think, one possibility. In ABC analyses, we had just modeled population expansion, lumping all of these inland samples together. Yeah. So the other thing that could be happening, of course, is that it's possible that these things, even though they've expanded their geographic range, haven't dramatically expanded their effective population size. And so we don't need to parameterize it because genetically it isn't important to parameterize it. And so I honestly have no idea which of these is the most reasonable explanation. But what I do know is one, I think, benefit of doing this kind of model comparison is you give yourself the opportunity to be surprised. And it's very possible, in fact, I know that this is not a model I would have sort of drawn down a priori as being a good fit to the data. But nevertheless, it's a model that has a high degree of support if we look at the IC differences in the model probabilities for these data. Questions about that? Thoughts about that? Yeah.
Do the willow trees reproduce clonally like some of the aspen populations?
They do to some extent. Our sampling was designed to, I mean, we can never be sure, but we don't think we sampled any clones. There's a pretty small geographic size that a clonal clump would have, and we were like several kilometers away even.
They probably can't clone like over a river or something. So.
Yeah, so there's that. But that's a possibility. Yeah. We tried to prevent that, but you never know. Plants are tricky. Salamanders are a lot easier in that way. So one thing I think that's worth noting about both of these tables is that our model probability between our best model and the other models for this data set is pretty dramatically different, both for this composite likelihood approach and the migrate approach. We have a smallish data set by next-gen standards here. There were something like 100 SNPs or something like that in the data set. So this, I think, is encouraging for people who want to use these approaches in general with next-gen data. It might not be the case that we need hundreds of thousands of SNPs to say something fairly definitive. Okay. So, I mentioned that there's a couple shortcomings of DADI. The dimensionality is an issue both because you can't compare more than 3 populations, but we also can't do something else that would be really useful. We can't compare a model that has 2 populations to a model that has 3 populations. Mathematically, those aren't comparable. And so I can't compare for Salix a model where everything is a single population and expanding to that model where there's 3 different populations. And so this is a shortcoming, I think, of the method. And presumably, I think it's something that we can do in Frapple.
Couldn't you do tau of 0 between 2 populations in DADI? I mean, I've never used DADI, so I don't know. But so yeah, they diverged, but they diverged yesterday.
Well, no, because you're still calculating the allele frequency spectra from X number, and that changes, the dimensionality changes that way that calculation happens.
So then that goes for the program Multipop out of Jody Hayes' lab.
I don't know.
He also uses the AFS, right? I mean, as long as you're using the AFS, you can't compare one population versus 2 populations.
I don't want to say in general because I'm just not sure. Some of this is also related to the composite likelihoods and how they're calculated too. And so I'm not sure where one ends in the other. So I do know that these AFS methods are increasingly popular because they allow us to analyze these big next-gen data sets. And so, Trace, what was that paper we just read out of Escoffier's lab?
Yeah, I'm trying to remember the name of the program.
So there's a new program that's coming out of Escaffier's lab that is similar to Dottie, claimed to be better performing and more flexible.
FastSimCol2.
Is that a?
Is that a? No, it's, I think it.
I think it uses FastSimCol2, but it has some other name. But anyway, so I think these are definitely something to keep an eye on. They might be really useful for a lot of us. Okay, so just to conclude in a few things, and then I will stop on time here. I think that our best argument for model selection is that it allows us to, in general, identify models that are a reasonable fit to our data, but it also allows us to understand something about our data and the information that we have. So this is a table from another paper that I did a few years ago where we had a smaller data set, something like 3 or 4 loci. And we used this model selection approach, and we found several models that had model probabilities that were high enough where we wanted to pay attention to them. And I suspect, and we've done a small number of exploratory simulations in our lab that seem to support this, there's definitely a relationship between how much data you have and the magnitude of the AIC differences that you're getting. And so I think that as we gather more data, there'll be more compelling reasons to use these approaches. The downside, of course, is that as we gather more data, if we're using programs that take increasingly longer to run as your data sets increase in size, and we're analyzing them multiple times for different models, and that makes the computational intensity a lot higher in general. And so again, parameter estimation, I think, should be secondary to model selection. And we should choose models, estimate parameters under these models, and then do model averaging. I also think when we think about model selection, it's important to recognize how widespread this has become in a relatively short period of time. So if we think really loosely and sort of in general about model selection for phylogeography, certainly we all know that lots of people do it in ABC and using ABC approaches. You can think of species limitation, things like BP and P, as being model selection, with these reversible jump algorithms that choose models of splitting versus clumping, essentially. And certainly you can use Dottie and IM. N has this now. Things like MS Bayes and even things like structure and Structurama on some degree are model selection. And so hopefully this isn't just sort of a historical accident, but this is something that's going to become more pervasive in the future. Yeah.
It is FastSimCol2.
It is okay. So the program out of Escaffier's lab is, I guess, FastSimCol2. So other questions here? I've meant to put an acknowledgement slide and I didn't, but we need to acknowledge NSF for funding. We'll do that. It'll go up on the website. So, well, thanks then. Yeah, Brian.
I have a question about the earlier part about how biologists are Bayesians or frequentists or likelihood. I mean, my impression that biologists sort of most biologists, like Clayton aside, don't care that much about that level of thinking. They care about, like, I want to understand what's happening in my system, what tool lets me understand what's happening. And some tools require prior, some tools don't, and you'll adjust. I mean, and does Sober talk about that at all, or is it?
Sober does, and I think the kinds of, Sober is not talking about phylogeography, he's talking about important things in his mind.
Yeah.
And, so what is the evidence for evolution as a theory in general, and how can we convince members of the public to believe it in general? And so I think, I don't know how important these sort of, you could characterize these as philosophical distinctions, but I think they influence how we analyze our data. And so often in subtle ways that we might not recognize. To me, the big difference is the difference between hypothesis testing, which tells you something on an individual model by model basis about the models you're using. It's either one that you can reject or fail to reject. But if you fail to reject 2 models, you can't evaluate which of those is better. That's a big difference between that and the kind of multi-model inference that you could do with AIC and information theory or Bayesian approaches. So do people care about philosophical differences? We have hardcore Bayesians in this room who would never use a non-Bayesian method. I say, how many people, probably almost everyone in this room was trained in an environment where Bayesian methods were pervasive for phylogenetics and population genetics, right? They weren't always. I mean, 2000 or so was, you know, not that long ago, and when I was a master's student, none of these methods were sort of out at that point. So Bayesian methods were, you know, they were the hot new thing in 2002, and I remember listening to certain advisors freak out about them. So, yeah, so I don't know. I mean, I think I'm practical.
Yeah.
But I think the cool thing is we have a lot of choices now, and we will have more in the future, the very near future. Yes.
Apart from other shortcomings from ABC, we can think about ABC and Frapple as they basically differ by the way you think, by what would describe your data better, either topology or summary statistics. Can you think in any case for which summary statistics would describe better some data set than topology?
Well, like branch length information on the gene trees is stuff that we're not looking at. I mean, if you had a method, which you could do with approximate likelihood 2, we don't, but you could do that.
So like MS Bayes is a method that, you know, you could imagine some version of MS Bayes maybe. This is the testing for simultaneous divergence that Mike Hickerson has developed and colleagues. You could probably imagine something that was looking at reciprocal monophyly of gene trees across some barrier in a bunch of different species. But it's the branch link information that allows you to really make some statement about the simultaneousness, or try to make some statement about that. So, you know, there's a case where they've done a lot of testing of summary statistics and convinced themselves that pi and pi within, pi between are the way to go. You know, so I think it's naive to think that there's a magic bullet for every possible scenario. There's some problems with summary statistics, and one is that you have to divide your samples into populations in order to calculate, you know, something like pi within a population. You have to have a population first, whereas a topology could conceivably, even though we use that population information, what I can imagine doing with Frapple is, because the topology is the same, we can change that mapping file of individuals to populations in order to make some evaluation of whether population assignments are reasonable or not. You know, so we talked about bootstrapping earlier. A cool thing to do with Frapple would just be to do bootstraps based on population assignment and see if your model probability for your best model changes. I mean, you would expect it to change dramatically as you got worse population assignments, maybe by picking at random or something.
That's what Brownie does for species dilemma, basically.
Right.
Yeah.
Maybe the migration rates. But I was actually thinking in cases more related to population genetics, like cases where you have really short, really recent divergences, maybe, that some summary statistics might be better, might describe. I know some cases in which what actually might help is out of frequencies. I don't know.
I mean, I think that's reasonable.
Yeah.
Although it's easy to think that gene trees don't give you information about really recent things because they haven't had time to sort, but there's still patterns there. There's still gene tree probability distributions. You know, you might not be able to look at an empirical gene tree and see it, and you might not have enough SNPs to actually recover the nodes that are important.
Okay.
But in theory, there's still information.
Mm-hmm.